Hello, everybody. I am Damla, PhD student, and I am working on regenerative medicine these days. And today our guest is Professor Yüksel Durmaz. Could you introduce yourself, Professor?
Sure, thank you very much, Damla. As you say that I'm Yasemin Yüksel Durmaz, the professor of biomedical engineering department in our university, and I'm chemist by major, and so later I focused on polymer chemistry. Then in my postdoctoral research, I applied this polymer chemistry knowledge to the nanotherapeutics that now currently I work on the polymer nanotherapeutics that can actually treat something in the body and whatever the problem exists. And so trying to develop a spatial drug delivery system, some tissue ablation system that unwanted tissue can ablate it through ultrasound energy, maybe some blood pressure regulation. Those type of work currently we focused on.
Okay, now here we want to talk about chemistry Nobel Prizes. As you know, it is awarded to click chemistry and bio-orthogonal reactions. So we can start with maybe what is click chemistry? Could you explain us what is it?
Sure, of course. Click chemistry actually is a concept, and so it covers kind of reaction series that they effectively react and very fast and selectively react. And so, of course, there are a series of reactions that meet the requirement of the click chemistry that I will explain what it is. With an example, for instance, in the chemistry, if you want to react A and B, just let's say these are any two compounds, to obtain the C, most of the time you won't be lucky to just get the C. And you may have C in the system, you may have A, you may have B, and some unreacted or side reaction that you don't even have an idea what are they. And so most of the time this is a challenge for chemists to purify the C from all these systems. So click chemistry actually is a kind of the reaction that says that when A and B react, that reaction yield or the efficiency happens more than 95%. They just form the C, there is no side product, and it happens pretty fast. And so in here, in the click chemistry Nobel Prize part, they appreciate much more on azide and alkyne click chemistry. So in here we can say azide is A, that has the 3 nitrogen in the structure, and the B is the alkyne in here that has the 2 carbon with triple bond. These two compounds, as pretty small actually, but you can conjugate them to anything even bigger to combine, and they react with each other, they love each other a lot and react very efficiently and fast without any side product. And so this is, we can say this like so like a 2 Lego pieces. They are perfectly fitting with each other, and when you put them, it just clicks like a snipping molecules together. So actually this azide and alkyne chemistry is not something new for chemists. We have known it actually since 60s that we had just the alkyne and azide, but the reaction was just regulated by the temperature. It was non-selective and giving the different kind of product. And when Barry Sharpless, one of the winner, introduced this concept as a click chemistry, he was actually referring a reaction that, especially azide-alkyne reaction, that will just react with the high efficiency and selectively. I have heard his motivation was saying that just think that you have ocean on the floor and oxygen at the top on the sky, and just as a chemist, imagine a reaction that does not get bothered by the water and neither the oxygen. So I think this is something amazing for chemists. And he initially, 2001, I think, introduced the click chemistry concept as a very efficient reaction. And then, meanwhile, another winner, the Morton Mandel, he was working another system that catalyzed by the metal. And so actually simultaneously Sharpless and Mantel realized that if they use the metal in this system, they can make this azide and alkyne reaction pretty selective. It just gives you one type of product and very effectively and fast. That's the most important part. I think reaction happens in a minute. Just imagine that normally chemists, we wait minimum overnight. I mean, just days for some reaction. You wait it and you don't even get the pure product. And now they're talking about something happens very fast and with a high yield. That was something amazing at the moment.
Yeah, it is unbelievable.
That's right, and it created lots of impact, of course, in the chemistry field, that everyone just realized that there is a possibility to have a more effective reaction without any side product in an effective way. So I think just the click chemistry as a concept is this one, that you have two spatial molecules that they are highly reactive against each other and effectively they react, and reaction is really fast and doesn't give you any side product. It just gives you what you wish.
Amazing. So other key point is, I think, bio-orthogonal reaction. Yeah. So maybe we can jump into that point. What is it and what are the advantages?
So that's also needs to be understood to be able to totally evaluate this Nobel Prize. So maybe I can start with the orthogonality, because bio-orthogonal, you understand that it's related with the biological system. Yeah. The orthogonality is something you have two substrates that they are reactive to each other, but they only react with each other, that does not interest with any other substrate that is exist in the system. So there are well-known examples. For instance, our DNA, we know that adenine and thymine is a base pair, and guanine and cytosine is the other one, right? Yep. So even though these are favorable pair, even though there are other options, but it's not favorable. That's a kind of orthogonality. This is something like this. And in here, you can think that azide and alkyne is kind of orthogonal. They just like each other. So that this is the bio-orthogonality means this when it happens in the body and does not interact with any kind of natural component of the body, that is we call bio-orthogonality. So actually that's where Caroline Bertozzi comes into the scenario, because this is the section that he developed actually. So on her work, that's also an interesting story to understand the biological effect of the click reaction I would like to tell. And so she was working on the polyglycans. That's a kind of sugar that most of the time our cell surface expresses. And so they mostly focused at the moment that these polyglycans are highly expressed on the cancer cell opposed to the healthy cell, and they're highly branched on the cancer cell. And they also figured out that a kind of highly branched polyglycan structure prevents cancer cell detection of the immune cells. At the moment, they were just trying to figure out a kind of inhibitor that will help immune cell to recognize the cancer cell by playing with the polyglycans. But as the chemist, she was trying to modify the polyglycan, but she was trying to do this in body. And so at that moment, there was, of course, multiple problem going on. Just if you think, for instance, as a chemist, when I want to introduce or build a reaction, set up a reaction, I take a clean flask and put everything perfectly in it, and A is there, B is there. I set the temperature. I even arrange the stirring rate. Maybe I need a pressure special, I do that one. Or maybe oxygen I don't like, I provide an inert atmosphere. Or maybe water prevents my reaction, and then I make it dry, everything. And so can you imagine body as a reaction flask? That lots of things going on. There are a lot of compounds in it, and it's a water environment. So that's something kind of difficult. And so they were trying to kind of figure out a way that these modification can be possible in body as well. So initially, she kind of realized that this is going to happen through orthogonality, because selectivity is something important. Other than azide alkyne, we know different molecules that they are orthogonal to. For instance, aldehyde and amine. And so, but all our body, the protein all have the amine group. I mean, it's not selective. You can't just do any selectivity, create any selectivity in there. That was kind of difficult for them, and they needed something else. And later she figured out that if she can send to the body a kind of sugar derivative that has the azide group on it, like simply say azido sugar, for instance, what basically happens, the cell metabolize this and uses to synthesize the polyglycan. And naturally, you have azide functional cell that expresses the azido sugar in the polyglycan structure. So that can be used as a precursor of the click reaction, right? But that's not the end of the story, of course. That's just the azide-containing cell you have. You need another component. That's A, you need B. So she needed to introduce, of course, the alkyne in the structure. But at the beginning, we said alkyne and azide, they react in the presence of the metal. So copper, you can understand that pretty toxic that you can't introduce copper into the body. And so no, not at all. And so, okay, this is not gonna work, she understand, but something that they developed and designed a new alkyne that is a strained alkyne, means that still it has carbon with triple bond, but it's embedded inside a cyclic structure that's pretty unstable, but they're using this instability as advantages. It does not need the catalyst to react with azide. It just immediately reacts. And by playing with the structure of the cyclic ring, they can actually arrange the speed of the reaction. That's even the great tool. And so later they introduced this cyclic alkene, which they call cyclooctene, and by, let's say, fluorescently take cyclooctene or drug carrying one or a peptide carrying one. You're just sending, you already have the azide in the cell that's functionalized at the surface. You can just label it. That was amazing at the moment. Their initial work was with zebrafish. That's a kind of animal model that is transparent, that you can actually see what is going on in the animal. And they injected some azido sugar to the zebrafish, and they labeled their cell with the azide group. And later they injected cyclooctene alkene containing fluorophore, let's say with the red dye. And after a while, the animals start shining red. And later they injected azido sugar again, that they labeled the later generated or newly generated cell with the azide again. This time they send another dye that half of the animal starts shining green. So what this means, actually, you can watch the development of embryo in the system. Similarly, you can watch the development of the disease. Or in cancer, most of the group, they try to understand what's going on in the cancer environment, that these cells, some get modified. Initially, that has a different task, like some of the immune system. Later, they have a different task in the cancer development. So you can actually track those type of cell. And so that was something opened up lots of new area, actually, for chemists plus biologists, that we can ask new question, or we can investigate the new disease that wasn't maybe possible before by using this tool. This is actually a practical tool that you can really do reaction in the body without interrupting or creating toxicity to the body.
Yeah. So as far as I understand, it is possible to observe molecular reporter in the biological system without interfering with the system. So is that what is it mean?
Yes,
actually?
yes, it is actually. As long as you introduce selectively your azide or alkyne into the body, that because I would like to emphasize one more time that we don't have any compound in the body that has the azide or alkyne. I mean, that's something you introduce outside, and if you let body to metabolize it, whatever region you target, that will be your your reaction part, actually partner in the system, in the body. So this will allow actually to track the new molecule, to maybe label the new molecule, certain cell type, maybe treat them in a certain region. I mean, it opens up lots of new field or the research area.
Yeah, what I can think is I'm using axolotl as a model organism to trace the regenerating, and we can maybe we can use that system for axolotl even.
I agree. For the regeneration, it will be a nice point to watch.
Yeah. So we can come to therapeutic parts maybe. Is there any therapeutics that uses this chemistry, and what kind of diseases it's focused on?
So I'm pretty sure that there are lots of people, companies that they use this tool, the click chemistry is a tool to practically get something. And so, but if we focus more on the bio-orthogonal side, that as far as I know, that Bertozzi's group already has been running several companies for the different application, and they were trying to focus on immunotherapy and cancer treatment and some molecular imaging as possible. Also, selective protein degradation is possible. I mean, if you can label the protein selectively in a way that you can just degrade that protein, that maybe creates the disease, I mean, in a different way. Yeah. And so also there are some studies on the rare disease, on the neurodegenerative side, that they focused, and this disease mostly originated from the certain enzymes deficiency. And so, I mean, there are really cool things that this chemistry can be a tool, since it can be done in body as well. And so, but as I say, that I think most important part that I believe the past 20 years, this is what researchers also did. We asked the different questions that maybe wasn't possible to answer before. By using this tool, we ask them again, or we treat them differently this time, and that opened up or maybe answer new problems.
Yeah. So, what advances has click chemistry contributed since it was discovered in 2001? Is it surprising that it won the Nobel Prize, or is it too late? What do you think about this?
Okay, so let's start at the end. I don't think it is too late, never too late to get the Nobel Prize. And so, but I will touch that later again. So definitely it created enormous impact, especially on, of course, this is a chemistry event, but chemistry plus the polymer chemistry especially. So as a polymer chemist, I can have a little time to explain this part. In the polymer chemistry, actually, the reaction that I described at the beginning, A plus B forming C, is even more difficult. Why? Because polymer is a macromolecule made out of the covalently linked monomer, that actually you have something like a necklace, a long chain that is high molecular weight comparing to the small compound, and it was really difficult to make A and B combination because there wasn't almost a way to purify it. In the organic chemistry, when you react A and B to form C, even though you get side product, but there is always way to purify it. Even though it is painful, there is. But in the polymer, sometimes there is no way. And sometimes we have two different polymer, we want to combine them, but they are not compatible with each other in a way that how we prepare it. So with the click chemistry, practically it opened up lots of new strategy for us, that we started designing new structure that it wasn't possible before. And not only to synthesize them, also we got them in a more pure way to have well-defined structure for all of them. what this made, we started to be able to design some new macromolecular engineering, and I mean different architecture that was difficult to do before. This brought lots of new application. So polymer chemistry that actually has a big contribution on the drug delivery system and the tissue engineering the field. I can say that now we have the lots of possibility to prepare, tune the material in a smart way. I mean, we can make the material smart that can sense the environment. And click chemistry played a really important role in here to increase the possibilities. So I can say that, so I remember 2001, when it discovered, then I was doing my PhD around that times. And so I can say that we apply almost whatever we know before, we revisit it with the click chemistry, because they gave us an purer product, I mean the better product with the good properties, and we know the properties very well. I mean, we can well define it. That created lots of impact on especially polymer chemistry, that people apply different, develop, I mean, better product. Also, as we talk, the bio-orthogonal chemistry, that's it. I mean, as a side field, but as important as the other field, they all got affected each other, and that part also got developed a lot to develop the new treatment type. So, is it too late? I don't think, as I say that, it's too late to get the Nobel Prize. But in the chemistry, most of the time, the Nobel Committee, how they act, I mean, when something new discovered, they let that discovery to create their own impact. And so I think last year Nobel Prize was also same. I mean, that has almost the same time passed after its discovery. So that was a kind of the scenario existing in here as well. It was discovered, and more chemists contributed on this development and created a significant impact. But also there are other examples in Nobel Prize in chemistry. For instance, I think it was 2004, the graphene, maybe you heard that. That's, I think, that was just 3 or 3.5 years later that they discovered, they got the Nobel Prize, because committee appreciated that this will create a huge impact. And that discovery also, some people say accidental discovery, but in any way, there was definitely effort behind it, nothing accidental. And so they discovered it, and we know that perfect optical properties, I mean, mechanical properties, and really good physical properties. It's just a one atom thickness carbon layer with perfect honeycomb structure. And so now, I mean, also graphene got used in lots of fields, and after it recognized by the Nobel Prize. So 2 strategies exist, never late. I mean, but I believe that after this point on, there will be more studies and in click chemistry. I mean, keep going, that's for sure.
Yeah, I agree with never late part. Yeah. So let's come to our country. What kind of studies are carried on our country about click chemistry and bio-orthogonal chemistry?
So the copper catalyzed click chemistry, that's not going in the body, the outside, I can say that it's pretty common in the research field, that lots of chemists, scientists in our country, that they effectively use this technology to develop something. I mean, maybe 2 categories we can put to just to develop the new chemistry or new architecture in terms of polymer chemistry, and also they use this as a tool to develop something. For instance, my laboratory, and as I say at the beginning, I'm designing these polymeric nanotherapeutics, and most of the time they're complex macromolecules that I try to combine multiple properties in a way that they work in a nice harmony. So it's not that easy most of the time in terms of chemistry. So click is a kind of great tool for us to combine all of them. As I say that, I can do this reaction if I fish in water, if I fish in organic solvent, if I at room temperature, pretty fast. You can even do this at the surface, not soluble at all. I mean, that provides lots of opportunity for the material scientist who works on surface, the polymer chemist like me who tries to develop nanotherapeutics, and just the pure polymer chemist that trying to develop a new architecture. That's a pretty useful tool that has been using people use in our country. But on the bio-orthogonal side, unfortunately, I'm not able to say the same thing. The examples are not as high as the other one. And this might be certain reason, of course, that this bio-orthogonal molecule, of course, even though azide is simple kind of sugar azide, you can get it. And there is a created library that you can buy from the companies. But they're kind of expensive and difficult to synthesize, maybe them in a different way that you can tune the rate of the reaction. I believe that may be a kind of makes people to think second time. But I definitely believe that if you can build a good collaboration with a biologist and or chemist that they know the problem and they know how to fix it, to use bio-orthogonal chemistry, I mean, they can create a really impact, and that could be an amazing collaboration to increase the examples in our country as well.
Yeah. So this is the end of my question. So we can take some concluding sentences from you, if you want.
Sure. I mean, as a polymer chemist, that's something that I use practically in my my work. That is another appreciation, and the extra makes me happy that this chemistry got awarded. Yeah. And so, as a woman, also I feel really appreciated that get inspired. But there was a winner. Among other two guys, that is a lady. And so I think that's also amazing. In every Nobel Prize, they create awareness and impact for sure. And so this chemistry also still preserves lots of future application that will open up a new treatment option for human being and for society.
Yeah, thank you for your clear explanations. I learned a lot, so I enjoyed a lot.
Thank you very much, Damla.
I think everybody will enjoy for it.
Thank you very much. It was a pleasure for me.